Modahl about transcriptomics.
All right. Good morning. So I will be talking about, well, the title is Transcriptomics in Snake Venom and Snakebite Research. So my last slide will talk a little bit about applying these same principles to snakebite. So a brief overview of the omic universe, which Mike actually already gave you, and even some slides yesterday. So we have the DNA, which would be the genomics. We have the mRNA, so transcriptomics, and the proteomics, which was just covered in the last presentation. And to give you an idea of what this looks like, so on the level in relation to a venomous snake, I should say. So on the level of genomics, we have here a whole venom gene chromosome description where we have found, Todd Casto and team have annotated the prairie rattlesnake genome and found the location of all the toxin genes on which chromosomes. And so that would be a chromosome level view of toxin genes. So to look at the transcript, so this next image is actually a combination of the transcriptome and proteome. So you have here the bars, on my left, we have the FPKM, which was fragments per kilobase per million. So that's an abundance measure for transcriptomics. And so we have those bars representing the expression levels of toxin genes. Then we also have bars within those subset, those darker bars, and that's actually the proteomic quantification, which was just covered a little bit in the last slide. And so this is how all of these omic technologies fit together. And today I'll be talking about focusing on transcriptomics. Just to first give you an idea of the applications for transcriptomics. So obviously there's just the profiling, the general description of a venom gland transcriptome. So you're going to be describing the toxin gene expression, which also does give you insight on the venom composition for that snake. Also, as previously mentioned, Mike gave me a very good introduction. I build the databases that then are used to identify the peptide fragments from proteomics, from mass spec. And the transcriptome is quite unique in the fact that, say, you're working with a new species or even looking at venom variation and a certain population produces novel isoforms of certain toxins, or they're completely novel venom protein families. Without that venom gland transcriptome database that is used to match the peptide fragments, those fragments would be entirely missed because a lot of proteomics is done by peptide mass fingerprinting. So there are a few cases where it is de novo, but mostly it's matching against that database. So this is the example, and obviously not as much detail as the last presentation, but how I work how my approach is complementary to proteomics. And so I am doing transcriptomics, which is next-gen sequencing. So I'm sequencing the transcript. And so that is also important to note. So I'm looking at transcription, and then those transcripts are then translated, and then that is the predicted protein product from that. So there is additional step after I do the assembly and annotation, is I'm still working with the nucleotide sequences. And then they're translated to predictive proteins, and that builds a custom database that then can be used to identify different proteins for mass spec. So my approach is very complementary to other omic technologies. Even as I showed in the last slide, for genomics, it's complementary in the fact that it's pretty important information to know which genes are expressed, because there are definitely, considering the presentations from yesterday, many gene duplication events, and not all of those genes are expressed at the same level or expressed at all. So other applications is once you have that predictive protein sequence based on homology to other known toxins, you can kind of predict the activity of that sequence or potential active sites. Also, you can look at toxin evolution because you have the transcript, so you can see the mutation rate of that particular toxin because you can look at single nucleotide polymorphisms for that transcript, as well as doing selection analysis. So positive selection analyses are commonly done with transcriptomics. So you see this in as part of transcriptome papers, is they're also looking at positive selection pressures, for instance, which is found to be quite significant for toxin genes. They're one of the faster evolving proteins. And so usually are experiencing positive selection pressures, actually, as what was covered yesterday with the whole predator-prey coevolutionary arms race that is also happening. Another important thing is, say you have a toxin in pretty low abundance, say from a spider or scorpion venom even, and you want to really characterize what that toxin does. It's depending on the snake, especially even rear fang snakes too, it's very difficult to get enough material to then go on to characterize the activity of that toxin. So by doing transcriptomics and getting the sequence of that toxin, then you can actually recombinantly produce that toxin. And so that actually facilitates characterization of the toxins as well, and their functional activity. And even the last thing here is the structural insights. So obviously, once you have the whole protein sequence, you can use certain programs like AlphaFold is very popular here, and to predict that structure. And also, I very recently was involved in a project where it was pretty exciting that they were using cryo-EM to look at toxin structures, and they didn't know where certain alpha and beta subunit positioned. And based on my sequences, that I was able to help solve that structure that was generated by cryo-EM. So it's actually, transcriptomics can be used in a variety of settings, and that's why it's such an important field that I encourage everybody to be interested in. To give you an overview of venom gland transcriptomics, it's going to start with dissection of the venom gland, RNA extraction, so isolating the RNA, in particular, the messenger RNA. So if you remember back in general biology, there's a lot of different RNA, transfer RNA, ribosomal RNA, but it's the messenger RNA that's encoding that are the protein coding genes, transcription of them. We have RNA sequencing library prep. You have the sequencing technologies themselves, and so your RNA prep for the sequencing will also be, it needs to correspond with the sequencing technology you're going to be using. And I'll cover all of this in my next slides. And then at this point for the analysis, first of all, if yours is a new species that's never been looked at, it'll be a de novo assembly of the venom gland transcriptome. But if it is the case where a genome does exist and you want to look at expression of toxins, you can take your RNA transcripts or your sequenced reads and map them back to the genome to then look at the expression levels. But I'll also be talking a little bit about this in the next couple slides too. And we also need to consider, as what's been discussed, especially yesterday, with the variation in a population, just because you have a species genome, maybe not all the genes are there, that mapping might not provide as much insight. And then you have the data analysis, which is where you take all the transcripts, because you're sequencing all of the messenger RNA. And so some of those are for toxins. In fact, for the venom glands, toxins are the most highly expressed. They're, if you look at the top 100 transcripts with the highest expression, they're probably going to sit. Probably 70% of them are going to be toxins. They're highly expressed in the venom gland transcriptome, or the venom gland. And that's just because that's the function of that gland, is to produce toxins. But then you also have non-toxins, which are also pretty exciting, because to properly fold toxins, for instance, there's a lot of chaperone proteins, which we know absolutely nothing about for some of these chaperone proteins. And so there's also interesting other questions that can be used by data mining transcriptomes. And then you also, as I showed in the previous slide, can look at expression levels. And for most cases, your highest expressed toxin isoforms I see correlated with the most abundant proteins in the venom. Not always the case, but more often than not, that is the case that I do see. So just to detail a little bit about dissection of a venom gland. So I have a couple of different venom glands here. So we have example of Viperidae venom gland, gaboon viper, one from a lapid, that's Najacuthia, and a boiga, so rear fang snake at the bottom. And so yesterday, Steve Mackesy gave a little introduction about the differences between some of these glands too, because actually the muscles that are used to compress the gland are actually different, and sometimes entirely absent, like in the case of most of the rear fang snakes. And also to even where the accessory gland is placed. So what I'll be doing for this talk is just reviewing a couple considerations for each of these steps to produce a venom gland transcriptome of the highest quality. So as actually just covered is venom gland transcription peaks usually 3 to 4 days. So this is what studies have shown when they've looked at transcription of toxin genes. It's usually 3 to 4 days after venom milking. And so what is ideal for a lot of studies is if you really want to look at toxin expression, make sure you're actually looking at all the potential toxin genes. You'll usually milk that snake, and then 3 to 4 days after, then the snake will be humanely euthanized, and the RNA extracted, or the venom gland dissected and the RNA extracted from that. And this actually seems to also be variable for different snake species, I'll also say, and even the toxins, as Mike even mentioned. So I've looked at some of that. And so some toxins are peaking on day 2 even, and then some on day 4, and even variation between isoforms. So that's not always the case, but at least you can generally say that 3 to 4 days after milking, you will have high expression of toxin genes because that snake is trying to replenish its venom supply at that point. Also, I was really excited that this, Wolfgang mentioned this the other day or yesterday, is how many venom glands, so your N number. So he was talking about proteomics, but this also applies to transcriptomics. And actually, we're even more restricted because we have to unfortunately sacrifice the animal to get tissue. And so if you look through papers, ones that are probably 5 years ago and older, we really were just doing N of 1 for venom gland transcriptomics. And this is actually something that is changing. And so I really encourage that if you're thinking about doing venom gland transcriptomics, please make sure that you have a good sample number. And so 3 to 4 snakes is kind of becoming the standard, at least for high impact journals, is that's what's being required. And in the field of RNA-seq in general, if you're looking at something like differential gene expression, these numbers are 3 to 4 is usually your numbers of replicates that are required for other studies as well. But this is definitely a shift I've been seeing more recently here in these last couple of years, where it's becoming incredibly more difficult. You just can't get those lower N numbers published anymore in higher impact journals. Also making sure that your individuals are all from the same locality, because as we saw yesterday and even covered today, the variation between different populations. And so that is also important to take into account. What is beneficial, one nice thing is that a pair of venom gland will share, from a same individual, will share similar toxin expression. So that's been seen. Granted, it's only been looked at in a couple cases. I think this was the Eastern Diamondback rattlesnake, if I remember this publication right. But usually that seems to be the case. So I'm just saying, like, generally, usually that's what we see. Of course, there's always potential exceptions out there. Also, as just previously covered with the spatial proteomics, this also applies to transcriptomics, where we have expression of toxin genes, because obviously you're detecting the presence of those different toxins on a protein level, but this also applies to transcriptomics. And so we've seen this with single-cell RNA-seq data sets where certain toxins are expressed in certain areas of the gland. So this is something to take into consideration when you're dissecting the gland, that essentially, if you're really going to be looking at the full transcriptome, you're going to need that whole gland, and it's really important to break up the tissue quite well. And so we finely chop so we'll take the whole venom gland, finely chop it, and use that entire thing. But you also don't want to overload if you're using an RNA extraction kit or triazole. So you have to be careful about that. But it's very important to make sure that you're sampling across the whole venom gland. And also, our RNA-seq and transcriptomics in general is a very sensitive technology, because you're looking at gene expression. And so if you accidentally get muscle tissue in there, then you'll be looking at gene expression in that muscle tissue, which is not obviously going to be producing toxins. So it's very important that you're only getting the venom gland tissue. And so actually at the Liverpool School of Tropical Medicine, we just have a couple individuals that are trained to dissect out venom glands, for instance, to really make sure especially in the case of those vipers where you see so the venom gland is it's composed inside of almost like a capsule. You can kind of see it's the lighter color there, but then you have underneath it or over it, it's all overlaid with a giant, that's a giant muscle, the compressor muscle. And so it's really important that that muscle is removed, which in the case of vipers, especially these large vipers like Bitis, you really have to make sure everything, the venom gland is cleaned off quite well with other tissues. And there's the accessory gland, which in itself is quite interesting. And so we usually dissect that separately and do transcriptomics on that separately because it is a separate gland and not producing all of the toxins that a venom gland is. So what has also been seen, and I did some of this work during my PhD, is RNA is also present, mRNA toxin transcripts are also present in venom. So I worked a little bit on this during my PhD. Well, I actually shouldn't say a little, I worked a lot on it to try to get it all optimized because it is a more tricky procedure. The amount of RNA is quite low. And in fact, just general considerations that I've realized with my work, really using fresh venom is best. Yes, you can get those transcripts from freeze-dried venom, but they're really poor quality. And just to, I would just definitely use fresh venom, and the RNA isolation method is critical. So I found Trizol is quite good. And not only just myself, but a paper here just published recently also found comparing different methods that Trizol performs the best. And in general, Trizol is the best if you have so the yields are highest with Trizol. And so if you have a sample where that's a consideration where you're not going to have a lot of RNA present, it is the best alternative, the best for RNA isolation. And now to talk about RNA isolation, so there's a couple different approaches here. So I just mentioned Trizol. So Trizol is a reagent, and it's based on organic extraction. And so it's good, as I just mentioned, for samples with low yields of RNA. But one consideration with Trizol is the solvents that are used are hazardous. You do have to take precautions. There's like phenol and chloroform. And so you do have to make sure you're working under a fume hood, and considerations of disposal of those chemicals also need to be taken into consideration. So that considered, Trizol will produce the best yields. However, we found using kits, especially column-based kits, it's overall a better quality of RNA. You have less contamination, especially with Trizol. Sometimes it's really difficult to remove all of the solvents from that. And so you would get low 260, which I have in the next slide here, 260, 230, where it essentially detects solvent contamination. So that is the issue with Trizol that is avoided with kits. They're also more straightforward and very repeatable. So they're great for undergrad students. So I have my undergrad students working on that using kits. And they also avoid using those hazardous solvents, as I mentioned. But their yields are going to be less than Trizol. So when it comes to RNA quality control, so after you've isolated your RNA, so how do you know if what you've isolated is any good? Because RNA is quite fragile. So it is rapidly degraded because unlike DNA, which is, has the stable helix structure, RNA is just a single strand. So it's not as stable. It's also not meant to be, around forever. In fact, gene regulation, it's the transcripts are, one of the ways genes are regulated is by degrading the transcripts. And so therefore there are a lot of RNases present, even in our environment. And so you have to be very careful when you extract RNA using certain sprays like RNase away to eliminate all RNases. But once you have extracted that RNA, there are different values that are looked at for quality control. And the nanodrop will help determine the quantity and if there's any of that solvent contamination or protein contamination. So actually the 260, 280 is looking at if you have contaminating protein in your RNA. 260, 230 is looking at if you have any contaminating solvents, which, like I mentioned with Trizol, is usually the case with the carryover from the process. Also, you want to quantify how much is there. So the disadvantage of the nanodrop is because it's reading at 260, that will be both RNA and DNA that it's reading. And so if you really want to know exactly to quantify your RNA amount. The qubit is quite good at this because it's fluorescence based. It essentially has these fluorescent dyes that bind specifically to RNA. So that will allow you to quantify just RNA. And it's overall not too expensive to have these. I mean, it is expensive to constantly get these kits, but the kits are to measure over 100 samples. So that's kind of nice. So once you're invested, it's not too bad. And it's a much more accurate way to quantify RNA. And actually we'll do DNA and protein as well. So there's kits for that. And then the final thing, and the most important, in my opinion, especially if you're doing next-gen sequencing, is your RNA integrity. So you want to make sure that you're not just isolating a good amount of RNA, that you're also isolating RNA that's intact, that all your transcripts are intact and not fragmented. And so that can be seen on a tape station or a bioanalyzer. And so it operates in a method of capillary gel electrophoresis, where it's going to separate out the RNA. And there are a couple of very large RNA transcripts. That's the rRNA, the ribosomal RNA, and they actually form the two subunits that are the 18S and 28S, the two big subunits. And that actually makes up over 90% of RNA. It's the most abundant. And so when you run these gels, you actually see, if your RNA is preserved, you see those nice two bands for those two big ribosomal rRNA subunits. And if your RNA is degraded, which you see a little bit, I don't have the pointing here, but you can see these ones that don't have the nice bands. You kind of get smears. And so that kind of lets you know that this is a low quality. And this is assigned with these instruments an RNA integrity number. So that's what the RIN is. And usually when you're doing next-gen sequencing, they actually have a requirement. And it's usually a RIN of at least 7 is needed, that your RNA is high enough quality to then go on for sequencing and library prep, I should say. Library prep's the next step, and then sequencing. So to give you just a brief overview of library prep, also, I should note that your RNA-seq library prep is going to be dependent on your sequencing technology. And so here I'm covering for Illumina sequencing technology because this is the most common. It's the cheapest right now. It's the most accurate. Nanopore is definitely improving, and we'll see in the next couple of years, and I haven't evaluated it recently. I just looked at it about 4 years ago. So maybe that will become more commonplace in the future. So this is for Illumina RNA-seq library prep. And just to give you a brief overview, what happens is we just want the messenger RNA. So we capture that using beads that have poly-T tails, or poly-T sequences that then bind to the poly-A tails of the messenger RNA. So then you're just getting all of those transcripts. Then you fragment these transcripts, and this reason being is Illumina is a short read sequencing technology. And so therefore, to get the entire transcript, you actually have to break it up into fragments, sequence those individual fragments, and that will help you get the entire one. Then you do the de novo assembly. So that's what I'll be covering in the next steps. Also, with sequencing, it's done from DNA. So you actually generate complementary DNA. So that is DNA that's complementary to the RNA sequence. So at that point, you have DNA, and it's much more stable. And also part of the sequencing technology is you need to attach adapter sequences. And so part of this is done with the Illumina sequencing flow cell. So what happens is, with your library prep, these adapter sequences are complementary to those that are on the flow cell, and this allows it to attach to the flow cell in the sequencing by synthesis to proceed. And so therefore, as part of the library prep process, is we attach these adapter sequences. And also at this process, you can attach barcodes as well. And this allows you to what's called multiplex samples, because you can do this library prep for different venom glands, for instance, have different barcodes for each one. So they're all uniquely labeled, and then pool them together and sequence them all together. And so that will actually reduce your total cost by doing it that way, by that approach. So once again, with the RNA sequencing prep, consider the sequencing technology you would like to use. This general overview is for Illumina sequencing. I mentioned mRNA selection. There is also rRNA depletion. So I mentioned the rRNA is the most abundant RNA. And so you can just remove all the RNA and then just sequence prepare and sequence the remaining. This is I would say not recommended for snakes or non-model organisms, because the rRNA depletion kits are kind of designed for model organisms. So just be cautious of that. In fact, if you outsource your sequencing, a lot of these companies actually won't do rRNA depletion because they'll be quite nervous if it'll actually work for your organism. Also, you can get stranded information, and that is the direction of the sequence. So that's actually quite good for assembly. Other considerations is the size of your sequence length. So I mentioned it's fragmented. So it's usually fragmented for a size of 200 to 400 base pairs. But the sequencing technology is still much less than that. It's usually 50 to 250 is usually the range for Illumina. And so think if you want longer reads or shorter reads. And you can prepare, so you can actually do this entire process in-house. And it kind of depends on the number of libraries you're preparing, whether that is cost effective or not. I just gave a couple examples of libraries that I've used in the past. I highly recommend this NebNex library prep. That's been one of my favorites. And it's the kit's very straightforward, and they have all the reagents well labeled. But you can also outsource your library prep to companies. And in the past, this used to be quite expensive, but now is coming down in price. So just considerations with sequencing, I mentioned short and long reads, and this can vary on sequencing technology. So I have the sequencing technologies there. And PacBio and Nanopore are the longer read sequencers. They're actually the 3rd gen sequencing technology is what they're labeled as, and Illumina is referred to as 2nd gen. However, depending on what you choose, there are differences in accuracy. So Illumina is known to be highly accurate, one of the most accurate out of the 3 there. And that's the reason being is, I show an example of this here with Illumina. So I mentioned the binding of the adapter sequences. And what happens is Illumina creates a little cluster. They're just done through PCR bridging, PCR amplification. And so it creates little copies, all of the same. And then for the sequencing by synthesis, you'll see a fluorescence. So you can see the fluorescence, and each nucleotide has a different fluorescent color. And so the machine reads that, and that's how it determines what the sequence is. And if you have a little cluster of all the same thing, then you're getting lots of these bright lights in that same location. And that allows the accuracy, because essentially you're getting, you're seeing the same result consistently across. So that base pair call is more confident. And so that's why Illumina is noted for this. So once you have your sequences, there's 2 approaches. You can align them to a genome and look at expression levels of those toxin genes. Some things to take into consideration is if your species has an available genome. So sometimes, or most of the time, that's not the case, that the genome is not available. And also it's also it should be noted that just because a genome is available, all the files that are needed for certain analysis might not be available. So I agree with the statement that about databases, what can be uploaded, there are various files that are needed for certain programs, and sometimes not all of those programs are uploaded for those. And the same for transcriptomes too. So there's variation. Also, the quality of the genome assembly, especially when we're interested in toxin sequences or toxin genes, sometimes the quality of the assembly is such that not all toxin genes are there, or most often the case, they're not annotated. There have been several studies here recently that have really focused on looking at the evolution of toxin genes. And so they have gone through and actually in a lot of cases manually annotated those toxin genes. And so those are available for some species. And also considering the origin of the genome animal. So say, and most often the case, how much time do I have? Or I'm getting close. 50, until the end of the session with question. Okay. All right. So for, you can either align to the genome or de novo assembly. And considerations for de novo assembly is quality control. You have to make sure that your reads are of the highest quality. So there's certain programs to evaluate this, like FastQC, and also Trim-O-Matic will be the program that if you see low quality sequences that will actually trim to produce the highest quality sequence for assembly. Oops. For de novo assembly, there's actually a lot of different de novo assemblers that are available, but they are drastically different in their ability to assemble toxin transcripts. And to give you an overview, these are all of the ones I've seen. I actually have to update this slide with every time I do this presentation, just because even more and more are added. So these are all different de novo assemblers that have used. These ones are the generic assemblers, and there also been a couple of groups that have developed venom gland de novo assemblers. So those are specific for venom glands, because venom gland tissue is quite unique and high expression levels of multiple gene isoforms. But as I mentioned, there's variation in the ability of those different programs to properly assemble a transcriptome. So here I actually this is a couple years back now. So this is probably, so it's a little outdated. There have been version improvements for these programs since, but I evaluated several different programs and their ability to assemble metalloproteases from Crotalus atrox. And you can see in the red there is those are the ones that were not assembled, and the red ones or the green ones are the ones that were correctly assembled. However, what should be taken into account is that certain individuals have genes that are lost. And so actually when the Crotalus, so after I did that work, the Crotalus atrox genome came out, and they actually found that two of those metalloproteases were actually missing from the genome that were characterized on a protein level previously. And so it's possible that, actually it's likely that I didn't assemble them because those genes have been lost entirely for that individual or that population. So you'll need to consider venom variation, especially when using a reference genome, for instance. So maybe even going onwards, even when a reference genome is available, we still might need to perform de novo transcriptomes assemblies. So a couple of assemblers I like as Extender is I found to be quite successful. It assembles really abundant transcripts quite well. Trinity, it assembles the lower abundant transcripts. It actually really suffers from the ones that are quite highly abundant. So it's not going to assemble metalloproteases really at all. It really struggles with that one. Also, even some three-finger toxins. So be very careful with Trinity. And there's VT Builder, which does okay. Extender is still my favorite by far. So there's advantages and disadvantages of each different assembler. So what I've found is actually using multiple, then I just, get the advantage of each one, and then I can just remove redundancy using a program like CD-HIT. So that's been kind of my pipeline and approach for de novo assembly of venom gland transcriptomes. Another thing I want to talk about is once you have that assembly, you need to annotate the toxins. And previously, this was quite labor intensive, where we would search against a database similar to what's done with the proteomics, and then go through and manually, determine like, is this a toxin or not? Because you also have the non-toxins expressed. The transcripts are there also there. And so recently, I guess a couple of years ago, there is a machine learning algorithm that was produced for annotating transcriptomes. And I can't say enough great things about this program, ToxCodon. It actually will annotate all of your toxin transcripts, and it does so within about 10 to 20 minutes. So really, really fast. It doesn't require a lot of computing power, so you can actually just run it on your own PC, and it outputs files for each toxin family. So it's all separated. So you have all your metalloprotease sequences there, three-finger toxins, et cetera, and it produces, shows even your translation and those final protein products. Another thing to look at is making sure signal peptides are present in your toxins, because toxins are secreted. So we want to make sure that that is there. And then also recognize based on similarity to databases, just because a sequence is similar does not necessarily mean it shares the same function. So if it's characterized as being a cytotoxin in the database, say, just because yours is similar, doesn't necessarily mean it has that function. So it needs to always be tested and functionally shown it has that activity. And then RSEM is used for determining transcript abundance, usually reported as transcripts per million. So this is just a couple of figures of showing you what you commonly see. So we see for each isoforms, we have their expression levels, and then we can do the pie charts and actually compare the transcriptome to the proteome here. And to also show you what happens when you don't have a proper database, I worked with rear fang snakes and there wasn't a proper database when I first started for this, this Amazon puffing snake. And so just using public databases, what was available. This was my pie chart. So you see certain things like L-amino acid oxidase tends to be overrepresented, and that's due because that's a very conserved protein in many different snake families. And so that sequence was in the database, something similar. But my three-finger toxins, which were quite diverse and unique, there was nothing similar in the database. So using public databases, a lot of my three-finger toxin peptides were not identified. But when I used my transcriptome reference database, my pie chart gradually improved. And if we compare it against, if this is just done with size exclusion, HPLC, and I determine those toxins and based it on the peak area, you see that it's much more, it's matching my shotgun sequencing of the proteome much more closely with my custom database. So once you have all the sequences, you can actually then look at the diversity between them. And just, this is my last slide, I promise. Just my final, I'm currently getting involved in a lot of snakebite transcriptomics. And so what this is actually looking at the toxin pathology on snakebite victims. And so by taking tissue from snakebite victims or even mice that we are using in the lab, for instance, looking at dermonecrosis models, so the MND model for mice, I've been taking lesion tissue and looking and doing transcriptomics on that to look at what genes are being expressed during tissue destruction from certain cytotoxic venoms. And so that begins to give us some insight on what is happening, the toxin mechanisms of action. And this can potentially be used to compare different models, such as, like I mentioned, the mouse. I've also been looking at human skin biopsies. And so this looks at gene expression between those two models and comparing to see if our mouse model is actually relevant to snakebite victims and how we evaluate or the neutralization of toxins and what toxins are doing. And I've been collaborating with a lot of clinicians as well. And through that work, actually getting tissues from snakebite patients, either muscle tissues or lesions. And so therefore, this paves the way for looking at potential adjunct snakebite therapeutics. So we have therapeutics that neutralize the toxins, but we don't know a lot about what the toxins are doing, for instance, inflammation processes. And so by using transcriptomics, we can actually kind of figure out what different immune pathways are being upregulated, and maybe there's adjunct therapies like certain immunosuppressants that could then be applied, or even biomarkers to get an idea of monitoring tissue damage. All right, thanks. Sorry if I went over, I apologize.